Welcome to another VoiceThread presentation. In this one, we'll discuss several transposition mechanisms, including cut and paste. Transposition is a type of genetic recombination in which mobile genetic elements move from one DNA site to another DNA site. These mobile elements are referred to as transposable elements, or transposons, of which there are three principal classes. The transposons show very little sequence selectivity when inserting into a DNA site and can therefore insert into a functional gene and disrupt that gene's function. As you can imagine, transposons could also insert into regulatory regions of a gene and disrupt function at the transcriptional level. Movement of transposons can occur with duplication and without duplication. If duplication is part of the process, the organism will end up with two copies of the transposon after transposition. In the case of no duplication, the transposon will simply be moved from one location to another in the genome. Transposition occurs in all life forms, and it is estimated that 50% of the human and maize genomes contain transposon-related sequences. Transposons are divided into three classes based on their overall genetic organization and mechanism of transposition: DNA transposons, virus-like retrotransposons, and poly-A retrotransposons. The virus-like retrotransposons include the retroviruses and may also be called the long terminal repeat, or LTR, retrotransposons. The poly-A retrotransposons are also called non-viral retrotransposons. This diagram shows the genetic organization of each class of transposable elements. DNA transposons remain as DNA throughout the recombination process and move by cleaving and joining DNA strands. The retrotransposons use an RNA intermediate to move to a new DNA location. In the following slides, we'll take a closer look at these. This figure shows the DNA transposon element, which includes the terminal inverted repeats and the transposase gene. The terminal inverted repeats carry recombinase recognition sequences that are about 20 to 100 base pairs in length and allow for transposase recognition. The transposase is the enzyme responsible for transposition. Target site duplication sites are direct repeats generated during recombination. If a DNA transposon contains terminal inverted repeats and a transposase gene, they are autonomous and can carry out their own transposition. If a DNA transposon carries only the terminal inverted repeats, they are considered non-autonomous transposons and require a transacting transposase gene in order to transpose. This figure illustrates the genomic organization of the virus-like retrotransposon and retrovirus. The LTRs, which you can see here, are direct repeats and are the site of recombinase binding and action. These LTRs incorporate the terminal inverted repeats that we saw in the DNA transposon element and can be 100 to 5,000 base pairs in length. Integrase is the recombinase gene, and RT is the reverse transcriptase gene. The target duplication sites are shown as blue areas with the blue arrow. You can see these on either end. As we mentioned earlier, retrotransposons require an RNA intermediate, So RT is necessary to convert RNA into DNA. The main difference between a virus-like retrotransposon and a retrovirus is the fact that the retrovirus is packaged into a viral particle that can exit the host cell and infect a new cell. The virus-like retrotransposon can never leave the cell and can only move to new DNA sites within the same cell. This figure illustrates the gene organization of the poly-A retrotransposon, which contains distinct sequences, including the 5' UTR, the 3' UTR on the opposite end, and the poly-A sequence. The genes contained in this retrotransposon are ORF1 and ORF2. ORF1 codes for an RNA binding protein, and ORF2 codes for a protein with both reverse transcriptase activity and endonuclease activity. You can also see the target duplication sites on either end, but you should note there are no terminal inverted repeats. Both genes play an integral part in poly-A retrotransposon transposition. The poly-A retrotransposon can be autonomous or non-autonomous. Now we'll talk about the actual mechanism, which is the cut and paste mechanism. And this is a non-duplicative method where one DNA segment or transposon is moved to a new location in the DNA. The basic steps include transposase binding to the terminal inverted repeats of the transposon element. This binding forms a transpososome or synaptic complex, and the transposase can then cleave the DNA to form the transposon. The transposon attacks the target DNA, and the transposon DNA is transferred to a new location in the DNA. This figure goes into more detail and will hopefully illustrate the cut and paste mechanism. At the top, you can see the host DNA containing the terminal inverted repeats. The transposase, which is represented by these orange squares or rectangles, recognizes these repeats and forms the synaptic complex. DNA cleavage occurs next to excise the transposon, and this includes the terminal inverted repeats. This is the excised transposon. The transposon attacks the target DNA at the site of new insertion. The site of DNA attack on the two strands of target DNA is typically separated by 2, 5, or 9 base pairs, which is represented in this area right here. And the 3' OH ends of the target DNA is cleaved. This is represented down here, where your blue is your target DNA. This separation gives rise to 5' overhangs on the target DNA and results in the short target site duplications that flank the terminal inverted repeats of the transposon element. These gaps are filled in by cellular DNA ligase. And this is represented here. These are the ligation of the nicks, and this is the new DNA that's filled in. This mechanism cuts only the 3' ends, and the following slide will show us how the 5' ends are cleaved. The 5' terminating strands at the junction between the transposon and the flanking host DNA are referred to as the non-transferred strand. This terminology arises because the 5' ends are not directly linked to the target DNA during DNA strand transfer. We referred to this in the last slide as the 5' overhang. There are several mechanisms used to accomplish cleavage of the non-transferred strand, and this slide shows the TN7 Transposon method. In this case, an enzyme other than transposase, which is TNSA, acts like a restriction endonuclease to coordinate with transposase and remove the transposon from its original location in the DNA. After excision, DNA strand transfer takes place as usual. The method used by TN10 and TN5 bacterial transposon uses the transposase itself to cleave the 3' OH end, which are seen up here, of the transposon, and then also attack the DNA directly across from the cleavage location. And that's represented by these red arrows here. A DNA hairpin structure, which you can see here, is formed and the ends are opened by the transposase to create the typical double-strand DNA break. So here's the hairpin opening by the transposase. DNA strand transfer follows the formation of the double-strand break. The Hermes method is similar to the TN10 and TN5 method in that DNA hairpin loops are formed, but the loops are formed in the host cell's DNA, as you can see on this either end here, rather than the transposon. This method is very similar to VDJ recombination and might suggest that transposons were leveraged for use in vertebrates. In replicative transposition, the transposon DNA is replicated during each round of transposition. The transposon DNA can be seen up here. Similar to the cut and paste method, transposase is responsible for binding to the terminal inverted repeats and forming the transpososome. The target DNA is then cleaved at the 3' OH region, still similar to cut and paste mechanism, but this is where the mechanism deviates. In replicative transposition, the transposon DNA is not excised, but the 3' OH ends are joined to the target DNA by strand transfer to create a double-branched DNA molecule intermediate. So here you see the 3' OH ends joining to the target DNA on either end, and this is your intermediate. The 5' ends remain attached to the original flanking DNA, and you can see this attachment here. The DNA branches within the intermediate are similar to a replication fork, and the cellular replication enzymes are capable of replicating the transposon and thus providing 2 copies. This method is prone to chromosomal abnormalities which can be detrimental. The last mechanism that we will discuss is using RNA intermediates, which are not as prone to error or disruption in the genome. The insertion of retrotransposons is very similar to DNA transposons, except that they use an RNA intermediate. The virus-like retrotransposon DNA element is first transcribed into RNA by a cellular RNA polymerase. So this is where you have your RNA element after transcription. The promoter sequence of the RNA element is found within one of the LTRs. The RNA element is then reverse transcribed into cDNA, and this is your cDNA. and a double-stranded DNA fragment is created which does not contain any host flanking DNA. The cDNA is recognized by integrase, an enzyme similar in function to transposase. Integrase is responsible for 3' OH-N cleavage, which you can see here, and DNA strand transfer. And this indicates the cleavage of the attack of the target DNA. Host cellular enzymes are responsible for gap repair and ligation. So this process is very similar to the cut and paste we've seen before. As you can see from this overview of transposition mechanisms, there are many similarities between each mechanism, but also distinctions between the different types of transposons and how they move from host DNA to target DNA.